Can you hear me?
Yes, ma'am.
Yes, ma'am. Good morning.
Good
Yeah,
morning.
good morning. Shall we start today practical?
Keep moving.
Yes, ma'am.
Okay. Okay. Today practical is the preservation of a specimen. In here in agriculture, we have to preserve specimens of insect, pest, and also beneficial insect, and also we have to preserve some disease plant leaf sample like that, so that today we will learn about the preservation techniques. What is the specimen preservation? Preservation is the long-term preservation of organism, either maybe it may be plant or animal, in the best possible condition. That is the definition of the preservation. Why we are going to preserve a specimen? Because we have to preserve that particular specimen for the scientific purpose. It can use as the reference collection in the future. That the specimens may be plant disease specimen, insect, or mites. I already told why specimens are preserved for detailed examination about sample and taxonomic reason for the zoological museum collection. We will see how classification of preservation. Preservation according to the two. According to moisture content, we can divide as dry preservation and wet preservation. Next one is the according to the time, we can classify it as short-term preservation and long-term preservation. For the dry preservation, best example is the herbarium. What is the herbarium? Herbarium is a collection of plants which have dried, pressed, mounted on herbarium sheets. After that, we identified and classified according to some approved system of classification. It is kind like a book. Uh, First, you have to collect that plant specimen and have to follow some steps: drying, pressing, mounting, and finally you have to label and classify. Is it understood?
Yes, ma'am.
Yeah. Uh, in herbarium, you have to, I told you have to follow some steps. No, first you have to collect the specimen. Next step is the pressing. Third one is the drying and poisoning. Next, mounting and stitching, labeling, and final one is the filling and deposition. Now we will see one by one. What is the collection? Collection means according to your objective, you have to go to the field. Then you have to observe. You want to observe uh, what are the species you want to collect. Then you have to collect and bring it for the pressing. That according to your objective mean, if you want to prepare uh, if you want to prepare weed album, so you have to collect weed sample. You have to observe that plant should be all the parts like leaf, flowers, or the root system. all the parts should visible in that sample. After collection of the sample, you have to press. Pressing means that fresh material containing moisture. No, if you can't preserve because of presence of moisture, that specimens may be damaged by mold or insect like that. So that you have to remove moisture from the sample. So you want to keep it within a book page or newspaper page like that. So after pressing, you can obtain this specimen with drying condition. Next one is the drying and poisoning. For the drying, you can use artificial heat or it naturally also you can dry with so that you have to use blotting paper or newspaper or something. Then that water from the sample evaporated to the that paper. Poisoning mean I already told some specimens may be destroyed by beetle or other insect. So that you have to uh, put some insecticide or fumigation of some chemical. So after you can preserve the sample. Uh, so overcome that insect problem, you can use some poisoning material. Next step is mounting and stitching. That mean fresh and dried samples you have to mount on the herbarium sheet for the preparation of the herbarium. Uh, as the mountain media, you can use sellotape or gum or something. Uh, Next, you have to go to the labeling. After mounting and stitching the specimen, you have to label because if someone observe that your herbarium, that person should get knowledge about that specimen. That specimen label should include botanical name of the sample, family, genus, species, local name, and date of collection, place of collection, and name of collected person. Because some specimens available only in one area, no. So that you have to write the place of the collection. Is it understood? All
Yes, ma'am.
Yes, ma'am.
okay. In here, you can see in this slide all steps in stitching and mounting, stitching, and preparation of the herbarium. I explained about the preservation of the plant sample in the dry preservation method. Now I'm going to explain dry preservation of the insect. Um, in here for the dry preservation, we have to use insect. They have a hard cover to dry in good condition. That insect only we can preserve in this dry preservation method. Because in in some area body is very soft body, and in some insect larval stages are very soft, no, that like caterpillar. You can't go for dry preservation because when we are drying that sample, it may be wilting, so that their appearance may be changed, no. So that you have a special kind of insect that have outer cover to dry in good condition. The samples kept in special boxes. That boxes are made from the woods and tightly closed. That box we are called as insect box. In this picture you can see this kind of insect box. In second year, third year, you have to prepare like this box also. And herbarium you have to prepare in one semester, I think, if for agronomy. After after drying, after killing the insect, you have to pin. Pinning mean you want to use pin to mount the insect in this box. Different insect have different pinning region in their body. You can learn in that semester. After that, you want to label the specimen. That the specimen with entomological name, date of the collection, place of the collection, like that. Is it understood?
Yes, ma'am.
Okay. You have problem, you ask me. Next one is the wet preservation. In preservation classification of preservation, we classified it according to the moisture content as dry preservation and wet preservation. Dry preservation we already learned. Now we are going to learn about the wet preservation. In dry preservation, I already told that dry preservation method is not suitable for the adult of some insect larvae and nymphal stage. So that we have to go for the wet preservation. And also that some insect have soft bodied and will easily decay insect there. For that insect also we have to go for the wet preservation method. In wet preservation we are using ethanol containers with a tightly sealed lid. And any soft-bodied arthropods we can preserve in this wet preservation method. And in here we have to write that label. We have to write with alcohol-proof pen if we are using this wet preservation method. In wet preservation, we have to we can use as the media alcohol, glycol like media we can use, and that is specimens may be visible as freshly killed insect or fresh sample. Like in this picture, you can observe that. Now we will see in wet preservation how to preserve insect in rubbing alcohol. Rubbing alcohol mean that normal alcohol we are using for the clean wounds or surgical instrument. By using rubbing alcohol, how to preserve insect? First, you have to fill that small jar or some that container you are going to preserve insect. You want to fill the container with half with rubbing alcohol. Next you have to find a dead insect, or if you catch one insect you have to kill. After that you want to find that dead insect and you want to label your insect for your sample. After that you want to carefully put the insect into the jar. Up we filled that half level of rubbing alcohol. Then we are inserting insect into the jar. Finally, you have to fill the jar, rest of the jar with the rubbing alcohol. After that, you want to seal the jar or container. Is it okay? Or I want to repeat it.
Okay, ma'am.
Okay, no? Now, we are going to learn how to preserve, how to prepare the slide for the entomological study. In here, we are using some common media for the preservation of arthropod. Here you can see some media like AGA solution, Barber's fluid, acid fluid, Hoyer's medium, and KAAD solution. Like this media we want to use for the uh, slide, slide preparation. In permanent slide preparation, we have to follow some steps. Here, first you want to kill the insect. Next, next step is the fixing and hardening. Third one is the staining. Fourth one, dehydration, clearing, mounting, labeling. Now we will see one by one steps involved in permanent slide preparation. First one is the killing. Here, if we catch some insect, first we want to kill the insect. For that, we are using some killing agent like 0.1% osmic acid, ether, alcohol, like that. A few drops of ether or absolute are placed on the slide over the object and are allowed to evaporate. Then that that insect will die. One is the fixing and hardening. Why we are going to fixing and hardening step? Because fixing steps stops any alteration in the tissues of the insect. It hardens the tissue and make them fit for extensive subsequent treatment. Because next we have to go some treatment. Before that, we have to harden the tissue and make a specimen with the slide. So that we are going to fixing and hardening. Here it makes the tissue suitable and susceptible for the action of stains and various other regions. In next steps, we are going to add some dye or staining agent. For that, we want to prepare. We want to prepare the specimen. Fixing agent make various constituent and component of a tissue that one optically differentiated by change in their refractive incidence. Here, we are using fixing and hardening agent. That agent help for the subsequent treatment of the treatment like the stain agent. Is it understood?
Yes, ma'am.
Can you hear me?
Yes, ma'am.
Yes, ma'am.
Okay. Third is in we are using dye that may be acidic, basic, or neutral dye. Why we are using this dye? Because that clear and absolute differentiation of the object. In this picture, you can see for the clear observation, we are using a staining agent or dyeing agent we are using for the clear observation of the specimen. Next step is the dehydration. Dehydration means the in your specimen may have a very small amount of water. If it dry the specimen, but that specimen may containing some traces of water in the tissues, so that we have to remove it and remove, and we have to replace by the alcohol. In here, the dehydration is achieved by passing the tissue through the gradually increasing percent of alcohol, like 30%, 50%, 20%, and 100%, like that. That dehydration means we have to remove traces amount of water present in the tissues with the alcohol. That alcohol may be different percentage level. Next one is the clearing. For the dehydration, we used alcohol now. In here, we in clearing no de-alcoholization. In here, we are going to remove the alcohol. Substitution of dehydration agent alcohol by the solvent found in medium is called clearing. That means we are going to remove the alcohol in the preserved material by using mountain medium. That mountain medium may be cedar wood oil or and clove oil, but in mostly we are use medium reagent is the xylene. That one is the mountain medium. By using this mountain medium, we are removing the alcohol presence in the sample. Next one is the mountain. What is the mountain? Mountain mean the in here we are using some mountain medium like Canada balsam dissolved in xylene and euperol. That one also we can use as mountain medium. How we are put this medium? Put medium in the center of the slide, and after that we have to cover with the cover slip. That means first we want to put that mountain media on the transparent slide. After that we have to cover with the cover slip. In here, in this picture you can see how to put that cover mountain media, and how to close that media with cover slip. That in here there's no there's no air bubbles present between the cover slip and the that mountain media. That our sample we have to put center of the cover slip. Then after we have to close the position. In first picture you can see that you have to close the cover slip with slant position, because if not that air bubbles may be enter into that mountain media, and that cannibalism or mountain media should not ooze out over the cover slip. You have to remove that excess amount of media presence on the side. finally we have to label the specimen, because after preparation of the slide, you want you want to observe in next in future, uh, so that you want to know about when you preserved that sample name, who prepared this slide, like that. So you want to label the slide in here. In picture you can see center of the slide have that preserved specimen. uh, both sides of both sides of the slide available some space. In uh, one edge of the slide you have to write about the specimen, that the specimen name, preserved date, like that. In another side of the another side of the slide you have to write about your details, that mean who prepared the slide, date of the preparation date, like that you want to write. After that you can observe, you have to put under the microscope, and you want to observe the microscope with um, suitable lens. So now is it clear to all how to prepare permanent slide?
Yes, ma'am.
Okay, is it okay to all, or I can repeat
Yes,
it?
ma'am. Okay, ma'am. Okay, ma'am.
Okay, no? Okay. Now, now we will see how to prepare temporary slide. Uh, ear- Earlier we learn about how to preparation of temporary slide. Now we are going to prepare temporary slide for the observation. That means we are going to prepare a slide for observation in temporary. That means that time we want to observe the specimen, so that to take clean slide, and you have to put drop of water on the middle of the slide. Excess, if they have excess amount of water, you have to remove that water by using tissue paper. After that you want to transfer material, uh, material mean that you want to observe specimen, you want to mount on the drop of water that you already put one drop of water now. You want to transfer observe and observe that observing material onto the drop of water by using a small brush. If they have extra fluid presence on the slide, you want to remove by using help of filter paper, and you can observe under the microscope. Like that you can prepare temporary slide for the observation. It is temporary. It's that slide we are preparing for the temporary observation, not preserved for the long time. This one, that observation in temporary time. Is it okay?
Yes, ma'am.
Can you? Oh, okay. Do you have any questions in today class? Do you have any clarification? You can ask me. Can you hear me?
Yes, ma'am.
Yes.
Uh, do you have any questions in today class? You can ask in if any clarification there. You can ask me. If not, I will ask some questions from you. I will ask randomly. Janani Upeksha. Can you hear me? Janani Upeksha.
Yes, ma'am.
Yeah. Uh, can you explain how we want to prepare a herbarium? Can you explain?
First, we have to collect specimen. Then we have to mm, flat. Then we have to dry them and
Yeah. First, we have to collect specimen. Second steps, what we have to do. Second one is the pressing, no?
yes, ma'am.
Okay. Third one, anyone
Dry
in your
precision
class
poisoning.
Trying and processing.
poisoning? Why we going for the poisoning?
Ma'am, to prevent
Mm.
the specimen from insects.
Uh, uh, we have to apply some insecticide because that specimens may be destroyed by beetle or other maybe any insect, so that we want to apply some poisoning agent. After that, fourth, what is the fourth?
Mounting and stuff.
And stitch.
Yeah, that what is the mountain stitching? Anyone in your class can explain?
Stitch the specimens by using gum or sellotapes.
Okay. Just after mounting and stitching, what is the next step?
Labeling.
Yeah. In label, what you write in label.
Scientific name,
Name.
place,
Botanical name, genus, family, date of collection, place of collection.
date of
Collected
person.
place.
Okay. Mm, why we are going for the wet preservation of insect or some insect instead of dry preservation? What is the reason?
Insects of good.
Okay. Okay. In discussion, you in examples of insect can preserve under dry preservation, and example of insect can preserve under wet preservation method. You want to search that insect example mean that insect name. You write in both method, dry preservation method and wet preservation method. Mm, any questions? If you have any questions, you can ask. Yeah. What? I couldn't hear you clearly. Okay, I will tell. In final one, that slide presentation, that picture you can draw. I will show. This picture you can draw. Um, um, someone asked why we can't some adult insect. I already told you. Dry preservation method not suitable for some insect. That insect have some insect have soft body, because in dry preservation we are going to dry the specimen. if insect have soft body condition, after drying, that insect may be wilting, so that insect shape may be changed, so that we can't use that insect for the dry preservation. Uh, like if some that insect have hard body outer cover, we can preserve that insect under the dry condition. Is it okay? Okay. Any questions?
Nilusha,
Yeah.
sorry for the interruption. Students,
Yeah.
students, if you have any doubts, you can ask in here. Then Nilusha miss clarify your doubts. Please don't keep your doubts in your mind. Some people ask the question related to previous practical question in the next practical. It not worthless, no? Please
Yeah.
ask the question in here. If you have any
If
doubts,
you
you can ask. Yeah.
yeah.
Students,
Yes, ma'am. Yes.
do you have any doubts?
No, ma'am.
No.
No, ma'am.
All of you understood the concept, isn't it?
Yes, ma'am.
Yes, ma'am.
Ma'am.
Okay, then okay. So if you have any doubts, you can ask in here. So in this practical, that is important. Okay, don't skip this practical. Each and every slide that is important.
If you have any clarification, you can ask me, because in next class, any other miss will take next class. No, then that you can't ask questions from them. If you have any clarification, you can ask today. No, it is okay. No.
Yes, in next practical, you may study the microbiological techniques. So if you ask the question related to this practical, the next practical will be interrupt. So therefore, please ask the question related to this practical in here. Okay.
Excuse me, madam.
Yeah.
Do we write the steps in passive voice or active voice?
Passive.
Okay, ma'am.
Passive voice. No?
Okay, madam. Thank you.
Okay. And
All the procedure should be in the passive voice. Okay.
if you have any clarification, you can ask. Now you have time to ask questions. No.
Okay. Shall I ask the question? The same question. Shall I ask? So any one of you tell why we go for the wet preservation other than the dry preservation? Why? Students, did you understood what I am asking?
Yes, ma'am.
So why we go for the wet preservation other than the dry preservation? Why? What's the purpose of that?
Because some insects have soft bodies. In dry preservation, we dry the specimens. So if we dry the arthropods who have soft bodies, their body shape will be changed and their parts will be withered.
Okay, can you tell some examples of the insect which may go for the wet preservation? Can you?
Butterflies.
Butterflies, we can go for the dry preservation.
Caterpillar.
Yeah, caterpillar.
Yeah, caterpillar.
Beetles, we can go for the dry because that is hard body, no? So we can go for the dry preservation. Other than lava, the caterpillar
Yeah.
also the lava, no? Yeah, the larval stages. Other than that one, sorry. Sorry. Can you repeat again? What's that? NYMPHS.
Nymphal stage, I think.
Nymphs. Okay. Yeah, okay. Nymphs. Some insect like whitefly, the soft body insect, thrips, whitefly,
Mealybug.
mealybug. Yeah, good. Mealybug, apit. We can preserve. Okay. Okay. So, what is that herbarium? What do you understand about the herbarium? Herbarium. Nilusham is also teach that one, no? What's the herbarium? And also she asked the question.
Yeah.
Yeah.
You have to prepare her. Yeah.
Labeling. The last one is a labeling, no? Yes. What are the material we can use during the mounting?
Gum, sellotape.
We can use some oil, no? What are those? Cedarwood oil, clove oil. Yeah. Then in one oil,
Xylene.
xylene. Yeah, good. Yeah, good. So good. Okay. I think all of you understood that practical. Good.
Yeah.
Yeah.
Do you have any clarification? I think all understood, no?
Yeah. I think so.